This is the review video for Lecture 4, Viruses and Prions. And this is a big lecture that starts with viruses. Viruses can be tricky to learn about because they are not like cells, but they are made of cell stuff and they are made by cells. So they are just a complicated thing to think about. And part of the challenge here is that you're going to come across some information that won't make much sense until you've gone all the way through the slides. So hopefully by the time you're watching this review video, you've done that and it makes some more sense. So we start with what is a virus, and again I show you a distinction between a virus and a virion, where a virus is a whole thing, like a whole plant, and a virion, the physical structure you see in diagrams like this, is really just better thought of as a seed that leaves, and all it can do is carry its genetic material with it, but it doesn't have any other functions at all. So thinking about viruses as these little structures misses everything important about viruses. You have to think of it, both these little structures called virions and the replication cycle, what happens inside the cell. That's a virus. So the first thing we do to look at viruses is we think about virions and the parts of virions. Okay, so what are the parts of a virion? It obviously has to contain the genome, which is a nucleic acid. It's either DNA or RNA, because that's its whole purpose, is to get that physical thing, that nucleic acid, into a new host cell where the host cell can use it to make more virions. It has almost always a capsid, which is a protein shell that protects the nucleic acid, made of capsomer proteins. So they are the proteins that make up the capsid. Some virions have matrix proteins that form an external shell even farther out than the capsid, and some have envelopes. These are roughly half of viruses have envelopes, very roughly. And then all virions have spike proteins of some sort, because the spike proteins are what attach to the future host cell. And they have to be very specific to attach to the right receptors on the surface of that host cell. And they have to do, well, basically that. So those are the parts of the virion, and it's a great idea to come back to this after looking at the replication cycle. Part of the challenge here is seeing the effects these all have, and the envelope is a big one. The envelope is an alternative place to route the spike proteins. So spike proteins for an envelope virion will be attached to the envelope as opposed to attached to the capsid in a naked virus virion. And an envelope mostly, well, it's usually a lipid bilayer, and it holds water in and kind of protects the virion by preventing it from drying out, but also it can act as camouflage. So make sure you review what envelopes are and what effects do they have. Well, they are going to affect how a virion penetrates a new host cell. That's going to be determined partly by whether or not there's an envelope. So envelopes are important. And then there might be enzymes and other stuff in there. So one good idea is to contrast virions with cells. So draw a diagram of a gram-positive bacterium, and you'll see it's surrounded by peptidoglycan. It has a thick shell, but it also, on the inside, it has DNA and RNA and proteins. A cell is getting energy from its environment, making ATP, driving all these intracellular processes. It's making its own lipids to make more cell membrane. Virions do none of that, okay? They don't make anything. They just exist, like a seed that does nothing until it germinates and by that analogy, germinating for a virion would be getting into a host cell and being disassembled. So there's that. A few other things we could think about would be the characteristics of viruses. And those characteristics are just the things we use to compare viruses to each other. I'm not talking about the symptoms and signs they cause. I'm talking about the viruses themselves. So each virus has its own tropism, and that's what kinds of host cells or tissues it can infect. Each virus has its own host range. That's the organisms, animals or plants or bacterial species it can infect. All viruses have a genome type. They might have double-stranded DNA as their genome, or they might have single-stranded RNA as their genome. So two of the most important viruses in the world, influenza and coronaviruses, have single-stranded RNA genomes. So there's no DNA in their virions. And no DNA involved in their replication cycle. They just skip all that. And remember that genomes can be, single-stranded RNA can be positive sense or negative sense. And that difference matters a lot to how biosynthesis works. But other than that, it doesn't matter much. That determines how biosynthesis is going to work, and it also determines whether or not there must be enzymes inside the virion. So negative sense RNA viruses must have certain enzymes in the virion, pre-made, before they go into a host cell. But we didn't get into that in this course. Another thing is viral latency. This is a characteristic some viruses have where they can hide in a host cell without replicating, without damaging it, without making new virions. That's latency, and we'll see there are a lot of human diseases that show that. Some other things would be, are they enveloped? What is the shape of the capsid? And then that's it for those characteristics. There's also virus phylogeny. And this is a diagram of phylogeny of some RNA viruses, specifically negative sense single-stranded RNA viruses. We can see that these viruses share a common ancestor, but we don't know if they have a common ancestor with positive sense single-stranded RNA viruses or DNA viruses. And none of them, there is no evidence at all that any of them have a common ancestor with a cell. So where did they come from? This is not known. This may never be known. But it doesn't seem like all viruses are related to each other, and they are definitely not related to us. They are made of the same general things we're made of, nucleic acids and proteins and lipids, but they're not made the same ways, and we don't know where they came from. So they have no place on the universal phylogeny. There are no viruses here. And their taxonomy is similar to the taxonomy of living things, of cellular life, but we just don't get into worrying about whether we're talking about a species or a virus or a strain. With a lot of viruses you'd care about, we just don't get that far into it. You don't need to know the strain of the rhinovirus that gave you the common cold, but obviously we know the sub-strain of the COVID that made you sick. We know the sub-variant that is circulating right now, and so that's way below the species, and it just doesn't matter what level it is. We just don't care about a phylum of viruses. We will often talk about viral families, and you'll see that later, but you don't have to worry much about that right now. So then we get into viral replication, and viral replication has a few big steps you need to know. You need to be able to list this. So these are the steps for a bacteriophage. replication cycle. It has to attach to the cell, and that's a very specific, critical process. If it attaches, it will probably successfully infect the cell. Then it has to get into the cell somehow. Then it has to force the cell to make new virion parts. It has to put them together in assembly, and it has to release them somehow. And so what these steps look like is determined by the host cell. So is it a bacterium that has peptidoglycan? That's going to affect penetration and release. What kind of genome does it have? That's going to affect biosynthesis. Is the host cell bacterial or eukaryotic? That's going to affect biosynthesis. But penetration and release are the most obvious things that would be different from virus to virus. And if we look at animal virus replication, they have another step, uncoating. And that's because their penetration involves bringing the entire capsid into the host cell, bringing most of the virion into the host cell. So then it has to break open somehow. That's uncoating. Otherwise, it's mostly the same. But again, since the host cell does endocytosis and exocytosis and doesn't have a cell wall, animal viruses can penetrate in several ways, and their release, they can be released without killing the host cell. So that's something. So again, the thing here is to think about what controls or what causes differences in replication cycles. Why is there an uncoating step in animal viruses? What is the effect of the envelope on how a virion penetrates a host cell? What is the effect of having a cell wall or not? Those are the things I want you to think about, because in a biology class, we are always interested in how does the physical structure determine the function and activities? What is the connection between the things you can see in that structure and how it's going to behave? That's what you learn in A&P. That's what we're learning here. So that's replication. And that's it for viruses. That was 13 minutes of viruses, and obviously it takes a lot longer than that to understand all these things. But this is just a review. Hopefully it refresh your memory a little bit. And from there we move on to prions. And prions, you have to learn this terminology. They are transmissible spongiform encephalopathies. And you have to learn the names of these diseases and the basic description of the diseases, and those descriptions come later in the slides. And then understand how TSCs work. How do prions work? And this is how they work. The way it works is PRPC is the normal version of the protein. PRPSC is the misfolded version, and if it is misfolded as PRPSC, it can change PRPC into PRPSC. So it's crazy. It's like once the protein misfolds, it can bump into other normal proteins and misfold them too, and then they can misfold other proteins. So it's basically turning into a catalyst of misfolding, and that spreads exponentially. So that's what you need to know about prions, that and the specific diseases, and why they're called spongiform encephalopathies. So that's the review video for Lecture 4. Lecture 4 is a big one, but at this point, most of you probably need to be focusing on all the other videos more than Lecture 4. So I'll see you at the next video.